Class, today I'm Harry, so we're going to have a roughly learning outcome for our new chapter. Today, we're going to learn about chapter 7. Recombinant DNA technology. Okay. In this chapter, we're going to have to learn about the definition of recombinant technology and technology, and list of tools used in recombinant DNA technology and function. List and function. Okay. The definition of the recombinant DNA technology is a method in which the molecule made in vitro segment from different sources or by combining genes from different organisms. Okay, tools that used in this recombinant DNA technology is tools. We have 5. 1. Target DNA. 2. Discern enzyme. 3. DNA. Joining vector. 4. DNA ligase. And 5 is host cell. For target DNA is a sequence of DNA containing gene of interest, while restriction enzyme is the enzyme which cuts DNA molecules at a restriction site and produce short restriction fragment. For DNA converter is a DNA molecule which can carry foreign DNA fragment into a cell and duplicate in that cell. For cell, a cell which able to receive DNA from recombinant allows the replication of recombinant DNA. And for modifying enzyme, we have 2 DNA ligase which join the foreign DNA fragment containing gene of interest and bacterial plasmid, and tag polymerase which used in polymerase chain reaction (PCR). For the second, the method for cloning vector for the recombinant DNA. Okay, first is isolation of DNA, isolation of gene. Then cleave or cut, insertion, transformation, and amplification. And the last is cleaning. Okay. Sorry guys, that's all for today. I'm in a hurry. I have to finish early, so I want you to please start to do your revision and please read all of it. Thank you.
Oh, Krishna, have you seen this cat that have been shaved by its owner? Oh, that's so cute. Yeah, they're so fun. I'm so hungry. Can I go buy something? Yeah. Let's go. Where you want to sit?
Just sit there.
Over 5. Do you remember?
I don't remember. Take out the...
Yeah.
Okay, so there are 5 tools.
Oh yeah.
Oh yes.
which is target DNA restriction enzyme,
DNA cloning vector, DNA ligase, and whole cell.
Can you explain to me about the target DNA?
Target DNA is the sequence. Here is the notes. Okay, the sequence of DNA containing gene interest. So the gene that they want, only the gene that they want is the what is target DNA.
That's all.
Yes.
We understand our food, so
Not really, but I can manage it. So the second one is restriction enzyme. And then this enzyme cuts DNA at the restriction site and translation or palindromic.
what do you mean palindromic?
Where the palindromic is where the same sequence of bases is found on both DNA strands but in opposite directions. So it's like this.
Yeah. Thank you.
What else? Sorry, buat saya tomorrow?
Isn't that Dia and Atika?
Oh yeah.
Hello guys.
Oh hello.
What are you guys doing?
We're just chilling.
Oh yeah. You guys know that we have to do presentation, right?
Oh yeah, it's tomorrow.
Is it okay if I one group with her?
Yes, we are okay. And we are in one group. How about we separate the topic?
Yeah, sure.
Oh yeah, wait.
You have the books? Oh,
Yes,
cool.
sure.
What topic do you want to do? I think. Did we take this right?
Yeah.
You agree with me?
Yeah.
Okay. Oh, okay.
Alright.
Cool. We're going to take this one, and also this one.
So we're going to take the rest, the three. This one?
Yeah.
Right. Okay, cool. That's settled. Where are we going next? Probably going to buy some stuff.
Yeah.
Okay. We're also better get going. It's going to rain soon.
Yeah.
Better hurry.
Alright.
Bye.
A few moments later.
Dee, isn't that sir?
Oh yeah. Hi sir, what are you here?
Oh, just read some recent news.
We want to ask you something about the presentation.
Oh, let me hear you. Do you have the end of the idea?
Oh yeah. This.
So, what you guys do?
We are going to do the target DNA restriction enzyme and DNA cloning vector. We don't know what else we must put in our presentation.
Oh, for target DNA, you can just deliberate. For restriction enzyme, you can just give the types of example cuts and the example of restriction enzyme. For DNA cloning vector, you can give the characteristic and examples of the enzyme.
That's all. Thank you,
Oh,
sir.
for the host cell and molecular enzyme and method cloning, we're gonna do it.
Oh,
Rahin and Husna.
Husna.
We're going to do it.
Okay.
That's all we want to ask you.
For your presentation.
Tomorrow.
Okay, this is our presentation today. Judges, please pay attention. Pause.
Assalamualaikum. Today we will present about recombinant DNA technology. First is to use the recombinant DNA technology. First is target DNA. Target DNA is sequence of DNA containing gene of interest. Example, gene encode for protein which can dissolve blood clot.
Okay, next, restriction enzyme. Enzyme which cuts DNA molecules at a restriction site and produce short restriction fragments. Restriction site is the short base pair sequence. Okay. Restriction site contains palindromic. Palindromic is where the same sequence of bases is found on both DNA strands but in opposite direction. There are two types of cuts, sticky end and blunt end. Example for sticky end is E. coli1, which is produced by E. coli. It recognizes the base sequence of 5' GAA, ETC 3'. It makes target cut within the sequence and it produces sticky ends like this. And for blunt end, it is example for blunt end is SMA1, which is produced by Serratia mercescens. Recognize the base sequence of 5' CCC GGG 3'. The cut is made between the adjacent C and G. Cut straight across the double helix and produce blunt end.
For the third rule is DNA cloning vector. DNA cloning vector is carry foreign fragment into cell and replicate into that cell. The characteristic is able to assert foreign DNA in MCS. MCS is multiple cloning site. Second, able to replicate freely in the host cell. And third is post-selectable genetic marker. Example for DNA cloning vector is plasmid, bacteriophage, cosmic, and YAC. The difference for the example is the size. The biggest size is YAC and the smaller size is plasmid.
So that's all from us. Thank you.
Okay, the next group. The others pay attention.
Okay, so we're going to discuss about the two other tools used in recombinant DNA technology, which are host cell and modifying enzymes.
For host cell is a cell which is able to receive DNA recombinant and allows the replication of recombinant DNA. Its characteristics is able to receive recombinant DNA through transformation process, able to maintain the structure of recombinant DNA from one generation to another, able to amplify the gene product from the recombinant DNA. Bacteria is the most commonly used as host cell. The reasons are DNA can be easily isolated from or introduced into a bacterium. Bacterial cultures grow quickly, rapidly, replicating any foreign genes they carry. Example, E. coli.
Okay, for modifying enzymes, there are DNA ligase and type polymerase. DNA ligase are joined to form DNA fragments containing gene of interest and bacterial plasmid by catalyzing the formation of phosphodiester bond to form recombinant DNA. For type polymerase, used in polymerase chain reaction, also known as PCR, replace the DNA polymerase because of the temperature conditions of PCR to add nucleotides to the 3' end of primer in order to produce new DNA strand.
So, the correct sequence in gene cloning, isolation of gene, cleave or cut, insertion, transformation and amplification, and also screening.
So for isolation of gene, plasmid from bacterial cell is isolated. Gene of interest is isolated from DNA of donor cell.
For cleave and cut, DNA donors and plasmid bacterial is cleaved using compatible and restriction enzyme. And also makes a single cut within the LEGZ gene, which is site of cleaving, producing complementary sticky end.
Okay, so for insertion, DNA fragment or gene of interest is inserted to the plasmid. It is also joined together by DNA ligase forming recombinant DNA.
For transformation and amplification, the recombinant DNA then inserted into the bacteria through transformation process. As the bacteria reproduce, the recombinant DNA is also cloned.
For screening, is to identify bacteria clone containing recombinant DNA. Blue-white screening is done. Plate the bacteria on agar containing antibiotic, ampicillin, sugar, and X-gel. Incubate until colonies grow. So that is all from us.
Thank you.
Thanks.